You know, in genomics, we're practically swimming in data. We run these massive experiments and get these huge lists of genes. But what if the simple, everyday tool we all use to make sense of it all has a hidden flaw? A flaw so subtle it can make us chase ghosts, celebrate false discoveries, and completely miss the real story. Today, we are pulling back the curtain on a problem called background bias.
So picture this. You've just finished a huge experiment. RNA-seq, proteomics, whatever your flavor is. And bam, you've got it. Your prize, a list of significant genes. It feels like a real eureka moment. But a list of names isn't a story. The real question, the one that actually matters, is what on earth does it all mean?
So how do we get from that list to actual insight? Well, to bridge that gap, researchers pretty much everywhere turn to this one really powerful computational method. It's become an absolute cornerstone of modern biology. It's the go-to tool for finding the biological narrative hidden in the data.
And that tool is called functional enrichment analysis, or FEA. The best way to think about it is like this. It takes your gene list and basically asks, hey, are there way more genes related to, say, immune response here than I'd expect just by random chance? It's a statistical test to see if there's a theme, a common thread in your data.
It is by far the most common way we turn a boring list into a real mechanistic interpretation. It's how we find the story. But, and this is a really big but, the very simplicity of this tool can hide a major pitfall, one that can send our research down a completely wrong path.
So let's get right to the heart of the issue, the bias in the background. See, the statistical magic of this whole analysis hangs on one single crucial choice. What are you comparing your gene list against? That comparison group is what we call the background universe. And this right here is where everything can go sideways.
As researcher Guangcheng Yu points out in the paper this whole explainer is based on, most of the software tools out there, they default to using every single gene in the organism's genome as that background. And at first glance, that kind of seems logical, right? But it's built on a really flawed assumption. Okay, so this slide just lays it all out. The flawed assumption is that every single gene in the genome has an equal chance of popping up in your experiment. But the biological reality? That's just not how it works. I mean, think about it. If you're studying a brain sample, you are not going to detect genes that are only ever switched on in the liver. Using the whole genome as a background, it's like trying to figure out if your friends are tall by comparing them to every person on Earth, including toddlers. The comparison group is totally wrong, so of course your conclusions are going to be skewed.
So what's the actual damage from making this flawed comparison? Well, it's that your big discovery might actually be a complete deception. You end up with results that aren't just wrong, they're so blindingly obvious they make the whole analysis a waste of time.
Let's go back to that brain sample. If you compare your gene list to the entire genome, the analysis will come back and triumphantly discover that genes related to neural pathways are enriched. But come on, that's not a discovery. Of course, brain tissue expresses brain-related genes. This kind of result tells you nothing new. And worse, it can totally mask the actual subtle biological changes you were trying to find in the first place.
And here's where it gets really dangerous. Using the wrong background doesn't just tell you something you already know. It acts like a giant megaphone for statistical noise, making what could be random chance look like some groundbreaking finding. It's basically a machine for generating false positives. And this chart shows it perfectly. Just remember, in science, a tiny p-value is what we look for. It suggests a result isn't a fluke. Now, look at the bar on the left. Using the whole genome as a background gives you this teeny tiny p-value. It's screaming significant. But now look to the right. When we use the correct background, only the genes we could actually detect, the p-value is much higher, much more realistic. That first result looks like a Nobel Prize, but really, it's just a statistical illusion.
But wait, because the problem actually gets even deeper. There are roughly 20,000 protein-coding genes in the human genome, and that huge number introduces a second, totally hidden layer of bias into the mix.
The problem here is something called annotation coverage. You see, for the analysis to even work, our genes need to have a known function written down in a database, like Go or KEGG. But look at this. About 30% of human genes don't. They're mystery genes. When you use the whole genome as your background, all these unannotated genes are automatically counted as not in the pathway, which completely warps the math and biases the results even more.
Okay, so we've established the problem is serious. It feels like the whole foundation of this analysis is shaky. But, and this is the good part, the solution is actually surprisingly straightforward. It's all about building a better, more honest background to begin with.
So how do we fix it? It's this beautifully simple two-step process. First, step 1, you build your background only from the genes that were actually detectable in your experiment. No liver genes in a brain study. Simple. Then, step 2, you filter that list down one more time, keeping only the genes that are actually annotated in the database you're using. That's it. This gives you a background that's relevant, fair, and just plain scientifically honest.
And you know what? Some tool developers are already way ahead of the curve. The R-package clusterProfiler, for instance, gets a big shout-out in the paper for being one of the heroes of this story. It smartly defaults to doing that intersection with annotated genes, which protects users from these common pitfalls right out of the box.
This all brings us to a much bigger point. Fixing this isn't just about one researcher changing one little setting in their code. We're talking about a systemic issue that really needs a cultural shift across the entire scientific community.
A survey of published research found a pretty shocking number. Only 4.1%. Just let that sink in for a second. Fewer than 1 in 20 papers even bothered to report which background they used for their analysis.
And the paper argues, really compellingly, that we cannot treat this like some minor technical detail. It is a fundamental choice. It's what determines whether your results are valid or just garbage. For way too long, the community has kind of overlooked this, and that's potentially led to countless misleading conclusions in the literature.
So what's the path forward? It really has to be a three-pronged attack. First, researchers. We need to be educated and take responsibility for defining an appropriate background. Second, tool developers need to build in safer defaults, just like clusterProfiler did. And finally, journals and reviewers have to enforce this stuff by requiring authors to clearly report their methods.
Ultimately, it all comes back to this one powerful idea at the heart of the research. The pathways we report should reflect biology, not bias. Our goal is to uncover the truth, and that means we have to use our tools for genuine discovery, not for distortion.
Which really leaves every single researcher out there with one final crucial question to ask themselves. Is your analysis revealing biology, or is it just revealing bias? I would really encourage you to take a hard look at your own methods, because the answer could change everything you think you know. Thanks for tuning in.